It was good. Today we got photosynthesis. Let's get into it. So what is photosynthesis? Photosynthesis is the process of using light energy from like the sun, photon energy, and transforming it into like chemical energy like glucose. Overall chemical formula, well, we got sunlight plus H2O plus CO2 turns into O2 and CO2. Not O2, glucose. C6H12O6 and O2. What is the light absorbing pigment? Chlorophyll. Anatomy of the chloroplast. So we start from the outermost. So we got the outer membrane, then we got the inner membrane. There's no intermembrane space. Within the inner membrane is the stroma. So that's like the kind of cytoplasm within the chlorophyll inner layer membrane. Then there's the thylakoids. There's the thylakoid membrane, which is like the thylakoid membrane is what's the thylakoid membrane is like what surrounds the thylakoid, right? There's the thylakoid lumen, which is within the thylakoid. And yeah, is that 8? Stroma. Stroma is that inner cytoplasm area, I'm pretty sure. There's no way it's... How is that? Wait, what? Yeah, that makes sense, right? Thylakoid lumen. What's that sound? Computer good, bro? Yeah, outer membrane, inner membrane, intermembrane space, stroma, lamellae, thylakoid. Then grana is a stack of thylakoid. Oh, why is this? Yeah, okay, stroma is just like the space within that inner membrane of the chloroplast. Okay. Where's the pigment embedded into? What is the structure of that pigment called? Well, it's embedded to magnesium, like in a pyral ring, like 4 pyral rings. So porphyrin, embedded into the thylakoid membrane is chlorophyll. So chlorophyll is in the thylakoid membrane. What are the most effective wavelengths of light? Red and blue are the most effective on getting absorbed, and then green is like emitted. What can photosynthesis be broken into broadly? So there's light dependent and light independent. The light dependent can be cyclic or non-cyclic. The light independent is just the calendar cycle. How many photosystem in light dependent? Difference between the two, primary rule of each. Okay, so photosystem 2 and photosystem 1. Photosystem 2 is PS 700, so it absorbs 700 nanometers of light. It's Newton meters? No, not Newton meters, nanometers, right? Yo, there's no way I'm tweaking about that, bro. So photosystem 2 is P680, actually I was wrong. Photosystem 2 is P680, so it absorbs 680 nm, and then photosystem 1 is 700 nanometers nm. I'm not sure if I'm saying if that's the right. There's no way, bro. And has I have this used to like being Newton meters, like Newton meters of torque. But yeah, anyways, P700 is PS2, absorbs 700 nm wavelength of light. That's like a key difference. What's the primary rule of each one? So photosystem 2 is mainly used for making ATP, while photosystem 2 is mainly used for making NADPH. What are the 5 steps of non-cyclic photophosphorylation? First step important. So the first step is that the electron gets excited in photosystem 2 and gets excited from photosystem 2. Where does this electron come from? It comes from photolysis. Oh wait, that's the next question. But okay, anyways, let me just talk about it more. The steps. So, bro, I'm so fried from today, bro. So, where was I, bro? Where did I even start? So step 1, electron gets excited by photosystem 2, then it goes over to the electron transfer chain, falls along the electron transfer chain, forcing electrons to go outside of the thylakoid lumen. Outside, right? Outside of the thylakoid lumen. Oh, it's from the stroma into the thylakoid lumen. Okay, so the inside becomes super proton. Bro, I should be knowing this, bro. Why am I tweaking out about this? Okay, let me restart. So first steps is electron gets excited by photosystem 2, electron then moves onto the electron transfer chain, then it moves down it and it powers active transport to pump electrons into the thylakoid lumen. Once we pump that thylakoid lumen out, once we pump the thylakoid lumen to be like, wait, what am I even saying? Once it's super concentrated H plus on the inside, then using chemiosmosis, H plus will go through ATP synthase and make ATP, and then that last electron will be accepted into photosystem 1. Yeah. So what are the final electron acceptor for after non-cyclic photophosphorylation in PS2? That'll be photosystem 1. What are the 5 steps of non-cyclic photophosphorylation in PS1? Okay, so first of all, the electron will get excited in PS1, then it'll fall along its own electron transfer chain. As it's falling around that electron transfer chain, it's going to make NADPH. That's just what happens. And then that last electron is I'm not too sure actually. Wait, did I talk about where the electrons come from? Yeah, I think I did, right? The electrons come from H2O, and that's how oxygen is formed. It's called photolysis. It doesn't talk about where it goes. It just says electron goes to photosystem 1, photons are excited to higher energy level, pass to the electron acceptor, travel down the ETC, and then the DPH is formed. That's the 5 steps it says. Not sure where the electron goes though. Like, does it just go to does it just hop on over to the oxygen or something, or what? Okay, anyways. Where does cyclic photophosphorylation occur? It happens in the stroma lamellae. What happens in it? Why does it happen? So when the electron is excited in photosystem 1, it'll be like, I'm not going to go to the photosystem 1 electron transfer chain and make any DPH. Nah, I'm going to go to photosystem 2 and I'm going to hop down that one and make some ATP. This happens because we need more ATP rather than NADPH, because sometimes in the Krebs cycle or whatever, we use more NADPH rather than ATP. So we use less. And wait, what am I even saying? Okay, we need more ATP in the Calvin cycle rather than NADPH. So if the Krebs cycle is like, yo, I need some ATP, like I've been using too much ATP and I got extra NADPH, then the cell will be like, I got you, like we're not going to do any non-cyclic, we're going to do cyclic photophosphorylation. We're just going to make some ATP instead of making NADPH that you don't need, you know what I'm saying? So yeah. Why is it called a light independent reaction? It just happens without light. Like you don't need light for the light independent reaction. Where does it occur? It occurs in the stroma. In the stroma. Yeah, it occurs in the stroma. What is the main purpose of the direct reaction? It's to fix inorganic carbon into organic carbon. What is carbon fixation and what organisms do it? Carbon fixation is just the process of converting carbon dioxide, an inorganic carbon, into a organic carbon. Autotrophic organisms do it. They just make their own energy. Word. Yeah, okay, carbon fixation, incorporation of carbon from atmospheric CO2 into organic compounds, done by autotrophic organisms, plants, chemoautotrophs, etc. 3 phases of carbon cycle. So first step, well, one step is reduction, the other step is, so the one step is regeneration, that's the third step. Second step is reduction, so the first step would be incorporation. I don't know, like, I know the first step, bro. The first step is just we make PGA, but what would you call that? Rubiscylation? Oh, it's just called carbon fixation. Carbon fixation, reduction, and regeneration. What are the 5 steps of the carbon cycle? So step number 1. Okay, so first of all, CO2 will come, and then a 5 carbon molecule called RUBP will come, and they'll get joined by Rubisco. That will form a 6 carbon molecule, because 5 plus 1 is 6. Then we're going to divide that into 2, and we're going to get 2 3 carbon molecules. These 2 3 carbon molecules are called PGA, right? Yes, okay, so 2 PGA are formed. Now these 2 PGA will get phosphorylated in the reduction phase of carbon fixation, and then we'll get G3P. Yes, okay, now after G3P, now what happens? G3P will move on to form glucose. Wait, how many steps did I even say? So I put 3 steps, so now G3P can either go and make glucose, or it'll go for regeneration in making more RUBP. Yes, most G3P is going to RUBP. How many cycles to form glucose? So we make from 1. Okay, so Rubisco, so we get 2 PGA. So what's the math? For 1 CO2 molecule, we'll get 2 PGA molecules, so that's 1 cycle. Glucose is 6 carbons. Glucose is 6 carbons. Is it 6 times? Oh yeah, 6 times to create 1 glucose. What was the reasoning behind that? I'm not too sure. Why is it saying 12 G3P? 12 G3P. G3P times 2. What? G3P. Oops. Wait, I just realized those messages show up. Bro, this isn't me, bro. It's just me talking, bro. Don't even matter. Why is it sick? Like, I just guessed it, bro. Like, kind of guessed it. So 1 CO2 will come. Yeah. Good night. I'm in the dining room table. Wait, so if 1 carbon. Oh, okay, so we you make two. Oh my god, bro. How do you know how many to how many? Where do we get 12 G3P from, bro? G3P is a 3-carbon molecule. We get a 6-carbon molecule, which is unstable. So we get 2 G3P per cycle, right? Yo, I don't understand this math right now, bro. 6 carbon molecules broken down to 2 PGA. That's 2 3-carbon molecules. PGA is phosphorylated to G3P. That's 2 3-carbon molecules. So most G3P is converted back to RUBP. Where are they getting 12 from? What the heck? I guess I'll just memorize the fact that there's 6 carbons, bro. Okay, what other molecule can interact with Rubisco? What is the relationship between that and the enzyme size 02? What is the process called? Another name. Okay, so this is called photorespiration. Oxygen can interact with Rubisco rather than CO2, and is the competitive inhibitor. Oxygen will just bind to the enzyme Rubisco and block CO2 from coming. 4 types of photosynthesis. So there's C2, C3, which is the normal one, C4, and CAM. Advantage of C4 photosynthesis. How does it do this? Okay, so first I want to say that C2 photosynthesis creates a pointless product. That's what happens when photorespiration occurs. C4, how does it do it? It like physically separates the location of like the light dependent and light independent reactions. Wait, yeah, so C4, so there's like the vascular tissue, there's the mesophyll tissue, and then there's the fibrous tissue or something. Yo, I'm so fried. There's a mesophyll, the bundle sheath, and the vascular tissue. So C4 photosynthesis will prevent photorespiration because the issue is oxygen, right? So you're using, so if you just like, if you keep Rubisco isolated, because oxygen is getting produced in the light independent reactions, so if you just split light independent and light dependent reactions, then you won't have the issue of photorespiration. So what C4 plants do is they'll do the light dependent reactions in the mesophyll. So we're gonna get the light dependent reactions are going to happen in the mesophyll cell, and then wait, what's going on here? Then there's going to be oxygen from that, but then we're going to have the Calvin cycle in the bundle sheath cell, so those are going to be physically separated, and then CO2 will just come in. I don't understand the function of malate though now. What does malate do here? Like, malate is... Okay, I think what's going on here is that... So, what I'm understanding from this is that the mesophyll cell is on the outer side, right? So that means that's going to be where the CO2 is able to come in from the stomata. So the CO2 is going to come in from the mesophyll cell, and then because we're not doing no Calvin cycle in the mesophyll cell, it's going to have to get to the bundle sheath cell. How is it going to do that? It's going to combine with PEP, form malate, and then malate will go to the bundle sheath cell, and then the Calvin cycle will occur, where CO2 will combine with Rubisco, etc. I think. Let me just like search this up. What is the role? Wait, what if I just do the AI? You very smart indeed. What is the purpose of malate in C4? acts as a carbon carrier. In seawater plants, malate transports capture carbon dioxide from the mesophyll shells. Okay, yeah, so the bundle sheath cells cannot capture carbon dioxide. That's why CO2 is able to, that's how you get CO2 to go to the bundle sheath cells, because it just can't go there by itself, because it has to go to the mesophyll. I see. Okay, so now the other one is called CAM photosynthesis. Oh yeah, 3 layers of these. There's the mesophyll, bundle sheath, and vascular tissue. What is the process of C4 uses? Different molecule. So it uses PEP to attach to CO2, and then that PEP CO2 malate combination will just go to the bundle sheath, and then that's where things will go down. Yeah, that makes sense, bro. Right? Now there's CAM photosynthesis, and this is different. Well, yeah, no shit, bro. No wonder it's different, bro. Why else would I be talking about it? What issue arises from it? How is it fixed? So, CAM photosynthesis is that the issue here is that H2O leaves from the stomata, so then it becomes dry in hot temperatures. So what will happen is that the stomata will close during the day, right? And the stomata will just be open during the nighttime. So now, slight issue. If the stomata, okay, so the light-dependent reactions will occur during the day. And during the day, we're going to form NADPH and ATP. That is mainly used for the Krebs cycle. So, what is the issue here? If we're forming ATP and NADPH in the light-dependent reaction, right? But keep in mind the stomata are closed during the day, then we can't do Krebs cycle because there's no CO2. So then, what do? We hold the NADPH and the ATP and store it into... Wait, hold on, I'm doing this wrong, bro. Okay, wait, I'm getting this backwards. We're not going to store the ATP. We're not going to store the NADPH. We're going to do the other thing. During the nighttime, the stomata are open. So CO2 can come in. But at nighttime, we're not doing light-dependent reactions. So that CO2 is just going to get stored in the vacuoles of these CAM plants. So it's going to combine with PEP, then we're going to form malic acid, and that's just going to stay in the storage vacuole. Then in the morning, when it's daytime, then the light-dependent reactions can occur. Those light-dependent reactions will occur, and then ATP and NADPH will all be formed, and then we can carry on with carbon fixation, because that malic acid can be then used for the Calvin cycle. Boom, crazy. That low-key makes sense now, actually. I was kind of confused, but yeah, that's basically all.